Hello, good folks, and welcome back to another rousing discussion of our flipped activity for A&P. So what we're going to be doing today is that we're going to go over Chapter 10 and Chapter 19. So I'm going to give you guys a moment and go ahead and pull up the Blackboard announcement that I sent you guys with these animations so they can be ready to go. All right, I'm glad you got those up. So we went over a lot of these answers in class, but just kind of give you a little bit more in-depth of what this means. So on page 1, part A, when it talked about the beta-lactam drugs affect bacteria but not human cells, what you read in your book and what you would have known going over the lecture notes for Chapter 10 is that those drugs only act on bacterial cell walls. They act on specifically peptidoglycan. So fungus don't have cell walls that are made of peptidoglycan. Plants don't have cell walls made out of peptidoglycan. Bacteria have cell walls that are made out of peptidoglycan. So that's why human cells are not affected by this particular drug. So that's why part A, the correct answer is letter D. Page 1, part B, Ben's infection is treated. A drug that's similar to penicillin is what treated the drug is what we call a semisynthetic. It means it's partially derived in the lab and it's partially naturally found in nature. So that's why that one is semisynthetic. On part C, it talks about the beta-lactam drugs affect the peptidoglycan layer of bacteria. Why would beta-lactam drugs typically be considered more active against gram-positives than against gram-negatives? Well, remember what we discussed way back in Chapter 4, I believe it was, about cell walls, and that the gram-negatives, they have a thinner cell wall, but they have that extra LPS layer that's around it. So as a result, sometimes it's a little bit harder, more difficult to get to the actual cell wall. So the beta-lactam drug has to get through that LPS layer first. Whereas with gram-positives, they just have that nice big thick cell wall. So there really isn't a barrier around it. So that's why letter D on page 2, the cell walls of gram-positive have a thick peptidoglycan content. All right, so we're going to get to our first animation now. So go ahead and pause this video and watch this very first animation. Welcome back. Wasn't that fun? So in part A, what is meant by selective toxicity? That is C. Chemotherapeutic agents should work against the pathogen, but not against the host. We should be able to select for what types of agents the treatment is going to work against. We don't want it to, and it depends. Sometimes if we don't know exactly what the causative agent is of the disease, we might want something more broad spectrum. But right now, if we're just trying to if we know specifically what it is that we are targeting, we want it to just select for that particular target and ignore other things. Part B, why are chemotherapeutic agents that work on the peptidoglycan cell wall of bacteria a good choice of drug? Well, we've kind of been talking about that all along these first 4 or 5 questions here, and that's because D, humans and other animals don't have a peptidoglycan cell wall, so that would be selectively toxic against those organisms that have a cell wall, namely the bacteria that could quite possibly be making us sick. Part C, why is polymyxin only used on the skin? You'll remember from the video. It can also, letter D, damage living tissue, cell membranes, but the drug is safely used in the skin where the outer layers of skin are dead. So if you had A&P, you talked about that, those different stratums. So we had the stratum cornea with the outermost layer, and if you had me for A&P, I probably told you that what people aesthetically love about you the most, your beautiful glowing skin and your long and shiny flowing hair, all that crap is dead. So that top layer of skin cells are dead cells. So this drug, polymyxin, could actually affect the cell membranes of not only bacterial cells but also of human cells. So we really only want to put it in places where those cells are already dead, so they don't, have the process isn't compromised for them. Part 9 here, what do these drugs act against, what bacterial target, and the target specifically that fluorochinones will act against are DNA gyrases, so it keeps that DNA from supercoiling itself back up. So what you'll probably found when you were going through the lecture and the notes were that antimicrobial drugs have a couple of major classes in which they act. They can act against the nucleic acids, they can act against protein synthesis, they can act against cell walls, they can act against plasma membranes. So some of these drugs have very specific targets. So for the fluoroquinones, their specific target is DNA gyrases. So we keep the bacteria from being able to replicate properly. Part E, why is it difficult to find a good chemotherapeutic agent against viruses? So this is right from the video as well. Viruses depend on the host cell's machinery, letter B. So it's hard to find a viral target that will leave the host cell unaffected. Typically, if we do something to affect the virus adversely, then it may also affect the host cell. So you have to be very careful on exactly what specific aspect of the virus that we are targeting. If we don't have that really selective specificity with antiviral medications, then the host cell will be compromised. Number one here, these are not from the video. These are just questions that you may have had to use your book for. Which of the following antimicrobial drugs would be useful to treat staph infection? One of my favorite antimicrobial drugs is erythromycin. These other drugs, some of them are deworming drugs. Some are made for other things, so this would have been just from your book. Part number 2, put the following route of administration in order, following the route that results in the highest concentration of drugs in the bloodstreams to the route that results in the lowest concentration on there. Letter B is the best answer of it. So the highest concentration of drugs we can get in the bloodstream is if we put it directly in the bloodstream. So that's what B is, intravenous. And then D, if we put it into the muscles, your muscles are highly vascular, which means they have a lot of blood flow that goes through to them. So if we give you an intramuscular injection of a drug, it will get into the bloodstream and you have a pretty high concentration of it fairly quickly. And then after that, we go to the oral route. Now it still has to go through the stomach and be digested in parts. Some of it, depending on what the drug is, can just be absorbed right through the stomach. And then a topical. Topical just means it's going on the skin. A lot of times topical drugs aren't able to get into the bloodstream at all, and if they do, they're in very, very small quantities. And we talked about polymyxin being a good topical agent to put on the skin because it has adverse effects on your cell membrane, so we only want it to be on dead skin. Number 3, if a particular bacterial species is resistant to beta-lactam antibiotics, which of the following might you expect to find inside the bacterial cell? So let's go back to what we already understood about those beta-lactam drugs, that they act against the cell wall, and we already understand that the cell wall is much more prominent. They're more peptidoglycan layer aspect, I should say, of the cell wall. In gram-positives and in gram-negatives. So we know how this drug works. So when we look at our options here, we have R plasmids, porins, egosterol, and resistance pumps. If you go back through your textbook, and then also just kind of using what we know that, all right, if some aspect of the cell wall that beta-lactams are working against, then what that can lead us to is the fact that there's got to be some sort of genetic change that keeps these, with the question specifically asked, that we would expect to find inside these bacterial cells. And what we expect to find would be these R plasmids. So that's the genetic change that we would expect to see. And number 4, a particular microbe gains resistance to gentamicin. You might also expect this microbe to exhibit resistance against streptomycin. They're very similar to one another. So that's letter C. All right, so go on to your next animation and pause the video here. All right, so part A, what is meant when a bacterium is said to be resistant to an antibiotic? Resistance means that the bacteria is not going to be affected by it. So if you're resistant to it, then letter C, the bacterium is neither killed nor inhibited by the antibiotics. No change is going to happen. Part B, when a patient is treated with antibiotics, C, the drug will kill or inhibit the growth of all of the sensitive bacterial cells. So letter B doesn't make any sense. That would kill or inhibit the growth of resistant bacterial cells. If they're resistant to the antibiotic, then they're not going to be killed on here. Part A, or letter A, mutations occur in all bacterial cells. Mutations can occur from the overuse and misuse of antibiotics, and then mutations are just wild things that happen. So that can happen, but we don't say that it's going to happen in all cells as a result of a patient using antibiotics. Sensitive bacterial cells multiply uncontrollably. That doesn't make any sense. If a bacterial cell is sensitive to this particular drug, then it will succumb to it. It will be killed by it. And then letter E doesn't work. Mutations will occur in all sensitive cells, but not in the resistant bacterial cells. If the sensitive cells become mutants, then they're probably not going to be sensitive to this drug or this antibiotic anymore. So the only answer that works is letter C. Part C, the process of acquiring antibiotic resistance by means of a bacteriophage. You don't even have to look at the answers for this one. Bacteriophage, remember those modes of horizontal gene transfer we talked about? Conjugation, transformation, and transduction. Transduction was involved a phage. That meant that a bacterial virus would take snippets of genetic information from one bacteria, the host cell, and bring it to another. So part C, correct answer is A, transduction. Then you have to look at those options. Part D, which of the following mutations would not result in an antibiotic resistance? Well, the only one on here that allows for the amino acid sequence to change, the only one on here that allows for the DNA to change but does not change for the amino acid to change is the silent mutation. So that's letter C. And then part E, our plasmids are most likely acquired through bacterial conjugation. And that. All right, so next video here, go ahead and watch it and then we'll come back. Part A, prokaryotes contain, and you didn't have to watch a video for this, the ribosomes are what size? 70S, yes, so letter D on page 6. Part B, antimicrobial drugs are selectively toxic. That means what? Does it mean B, the drugs will affect only a particular type of pathogen? No. It means that A, the drugs are more toxic to the pathogens than to the patient. That's what we're trying to select against. We don't want you to get sick. We don't want our patient to get sick. But we do want the bacteria to be taken care of. So letter A is the best answer as opposed to letter B. So letter B says the drugs will affect only a particular type of pathogen on there. That is looking at selectivity, but we are more concerned with it's you versus the bacteria in this aspect. So that's why letter A is the best one. Part C, the tRNA molecule holding a growing polypeptide chain is at the P site. And this is information that's straight off the video and information that we have talked about in our chapter on microbial genetics through protein synthesis. Which category of antimicrobial drugs works by changing the shape of a ribosome? And those would be those aminoglycosides. Those drugs are actually going to change the amino acid sequence, so letter D for part D. Which category of antimicrobial drugs acts to stall a ribosome as it reads messenger RNA? And that's just straight from the video, letter D, macrolides. And then part 8, not part 8, part F, the drug that blocks the action of a large 50S subunit. The way that this works, and this is straight from the video as well, it prevents the formation of peptide bonds. Part G, which antimicrobial drug affects the structure of the ribosome itself? Letter A, aminoglycosides, antisense nucleic acids. Those are all going to affect the structure of the ribosome because it screws with the nucleic acid sequence of what makes the ribosome. And letter H, which category of drug is complementary to messenger RNA of the pathogen? And that's A, antisense nucleic acid. So most of these answers, as you can see, came directly from the animation. All right, and for this one here, this is the animation on MRSA. When a nurse rubs your skin with rubbing alcohol prior to administering an injection, what process is he or she carrying out? Well, it's antisepsis. We're trying to kill any bacteria that would try to go into your bloodstream and cause sepsis. And it's also degerming, so it's wiping away those germs as well. Part B, disinfecting a changing table. Which of the following is not an important consideration when preparing to use a chemical to disinfect a diaper changing table in a public bathroom? A, whether or not hand sanitizer is available. That's not going to be a consideration. I mean, that might be a consideration while after you change a diaper or after you've cleaned the table, but whether or not what kind of chemical you would use, that's not going to play a role in that. Now, part B, whether or not the changing table is visibly soiled. That's going to determine what types of chemicals you want to use on there. Definitely you would want to degerm it, wipe up everything, but definitely since you've seen visible solids and visible soil, you'll want to spray something on it. Letter C, the label directions for contact time. So how long should you give whatever drug it is that you're using, whatever compound you're using on the label, how long should it be exposed to it? Is it for 20 seconds? Is it for 10 seconds? Is it for 20 minutes? Part D, whether or not the chemical is compatible with the material of the changing table. And so Part D and E actually are one thing. So some things are more porous than others, and if it's a very porous changing table, then you're probably going to want to give it a longer exposure time, and you're also going to want something that can get down into those little porous areas to get the bacteria where they may be hiding. If it's a much more solid and it's not quite as porous, that may also change the type of chemical that you would like to use. So then Part A, antibiotic resistance testing. Many MRSA strains are sensitive to other antibiotics, so they're not just resistant to methicillin, sometimes they're sensitive to other things. Which of the following method would be the best way to evaluate sensitivity to other antibiotics? And remember the Kirby-Bauer method we used in lab, that disk diffusion method, so that's why letter D is the correct answer. And then Part D, mechanisms of resistance. In examining a MRSA strain, researchers find that degraded penicillin is found in the culture medium. What is the most likely mechanism of resistance? And they're producing this enzyme that makes them resistant to that aspect of penicillin that targets cell wall, which is that enzyme beta-lactamase. So these bacteria will have this enzyme that will allow them to break down beta-lactam. Then the enzyme they have is beta-lactamase, so beta-lactam doesn't work well on it. All right. So here is the bottom of page 8, the correct answer for this part. And then for the concept map on the very last page, here are the correct answers for this. All right, so moving on to chapter 19. Sorry about that. All right, so in this chapter, and actually from here on to the rest of the semester, we're going to be talking about specific bacteria. Chapter 19 is dealing with gram-positives, and then chapter 20 will be gram-negatives on here. So for chapter 19, we're looking at all these gram-positives, and we're putting these appropriate pictures of into the correct spot. So we have actinomyces here, we have Staphylococcus, we have Bacillus, we have Clostridium on there that have the nifty little club heads. We have Streptococcus, and we have Mycobacterium. All right, so we only have one animation for this chapter. So part A, page 1, Streptococcus pneumoniae was originally named Diplococcus pneumoniae. Why was the genus name Diplococcus originally given to the organism? Well, you can probably figure some of that out just by looking at the name. So cocci definitely means that there were cocci or spheres that were associated with it. So we'll definitely want to find something in our answer that has something with spheres. And then diplo or di means two. So the correct answer, so there's only two of them that say anything about the prefix di means two. Okay. And these organisms are usually found in pairs under the microscope, and on agar plates, the colonies are found in two groups. Well, when we identify microorganisms and we're trying to describe their characteristics, we can talk about the characteristics of a colony, but when we talk about characteristics of the organisms themselves, we use the microscope to determine that. So that whole business with gram staining, and then I ask you guys, when I go around to look at your microscopes, what shape do you see, what color do you have? Those are all phenotypical characteristics that we can only really see under a microscope. So the best answer is letter B. These organisms are usually found in pairs under the microscope. Now this is information that you would have need to gone through your book to get. In fact, most of the answers for chapter 19 you'll have to go through your book in order to gets the correct answer. So then we move on to part A on page 2. The pneumococcal vaccine was made using purified capsule material from 23 common strains of Streptococcus pneumoniae, but it was not effective against all strains. Why is that? So what you'll find in your book is that a lot of these bacteria, even though they may be in the same genus or the same family, they all have some sort of differentiation to them. And that differentiation is determined by what types of antigens are on the surface of that cell. So if those antigens are slightly different, then the vaccine or the treatment may not work for them completely. So the best answer for that is letter D. It cannot cover all strains of Streptococcus pneumoniae because not all strains have the same antigens on their surface. So remember those antigens, and you've probably heard about antigenic determination in an introductory biology class or in A&P if you've had it already. Those antigens are kind of like those surface tags that will make one cell within the same species different from another. And the vaccines, if they're made to target and to seek out these very specific types of antigens, then if the cell is missing them or they're slightly different, it won't be able to detect them. Part A, based on its gram status, why are penicillin and other beta-lactam antibiotics effective against Streptococcus pneumoniae? We can go all the way back to chapter 10, you know, that happened like 10 minutes ago, and we can answer that question. So beta-lactams, they're going to work against bacterial cell walls. They work more readily against gram-positive bacterial cell walls. So let's find an answer that fits that criteria. Beta-lactam, letter A, beta-lactam antibiotics are effective against Streptococcus pneumonia because it is gram-positive and has an outer membrane. Well, some of that statement is true, it's gram-positive, but other parts of it are not true. It has an outer membrane. Do gram-positives have an outer membrane? No, so that makes that incorrect. Part B, beta-lactam antibiotics are effective against Streptococcus pneumoniae because it is gram-positive. All right, check. And it does not have an outer membrane. Okay, both parts of that statement are true, so that is our correct answer. But for poops and giggles, let's just go ahead and look at the other ones. Beta-lactam drugs are effective against Streptococcus pneumoniae because it is gram-negative. Yeah, screw that. Don't have to keep going on anymore on that because that's incorrect. Beta-lactam antibiotics are effective against Streptococcus pneumoniae because it is gram-negative again. We don't have to go with that one either. So our only correct answer is letter B. Next one. Strains of Streptococcus pneumoniae can cause disease only when a capsule is present. Ooh. So I want you to stop and think. Streptococcus pneumoniae, gram-positive, correct. Streptococcus pneumoniae, does it have an LPS layer? No. Streptococcus pneumoniae in this question is talking about can cause disease when it has a capsule on there. So remember the R strain and the S strain that we talked about with Griffith's experiment on transformation with those mice? This is the same drug here, same thing. So remember when it was capsulated, when it was smooth, that it actually caused disease and the mouse died? And we said the reason for that is because the capsule makes phagocytosis very difficult. So can we find something that says phagocytosis is difficult when there is a capsule present? So letter A says Streptococcus pneumoniae cannot adhere to the cells of the host without a capsule. Yeah, that's not correct at all. So Streptococcus pneumoniae cannot adhere to the cells of the host without a capsule. It totally can adhere to them. The capsule is for protection, not for entry. Part B, the capsule prevents bacteria from being digested after endocytosis in the cells of the host. That, on the other hand, is correct. And then let's just read C and D, see what they say. Streptococcus pneumoniae cannot migrate from the nasal pharynx to other areas of the body without a capsule. That's not true. That's not the job of a capsule. C, the capsule prevents the bacteria from releasing chemicals that announce its presence to the host immune system. Once again, that is not the role of a capsule. What this question really wants you to understand is what is the job of a capsule? And the job of a capsule around the bacteria is to prevent phagocytosis. Moving on. Part A on page 3. Streptococcus pneumoniae is found as a part of normal microbiota of the mouth and the pharynx, and yet can cause disease in some people when it is inhaled in the lungs. Why do you think this is? Well, this is information that you would have had to go straight out of your book in order to get. And our best answer for this is if Streptococcus pneumoniae is inhaled from the nasal pharynx and into the lungs, it could be damaged by the lung tissue can be damaged by a previous viral infection, or a person who has a compromised immune system will not be able to keep it under wraps, so that way it will actually be able to flourish. So really it has to do with the host being affected, the person being affected, and not necessarily with something being changed for Streptococcus pneumoniae. So correct answer is C. And once again, this is just a question that you would have had to just understand from your textbook. And now we have our one and only video. So let's go ahead and watch this video. And for this video, I think we're going to have to watch it together because I didn't send you all the link for it. And actually, I think I'm going to send you the link anyway because it doesn't, yes, so I will send you all the link for this one because it does not, the sound is really bad. So pause and go watch the video. All right, so on page 3, letter A. A characteristic symptom of necrotizing fasciitis is that the pain is A, disproportionate to the appearance of the infection. In fact, it is very, very painful, and so painful, in fact, but it just doesn't look like that. And I'm kind of feeling distracted right now because I'm trying to find a story that I just saw on the news about a little girl who had necrotizing fasciitis, or you probably know it by its street name as flesh-eating bacteria. And they had to amputate this poor little girl's leg. And they talked about her parents, and actually she had strep throat. And her parents talked about how when they looked at the infection, or they looked at the area, it didn't look bad. However, the appearance of the infection is much greater, or is reduced in comparison to the pain. And they think it started as strep throat. All right, and she was a little girl. She was only about 5 years old. So here is the news story for this. Oh, Christ, I have all these pop-ups coming up. So yeah, a 6-year-old girl in Ohio. She had to have her leg amputated after she had a complication from strep throat from this bacteria. So yeah, if you go to Live Science, and I will link this story into, I'll put this as a Blackboard announcement as well. All right, so part A, letter A is correct answer. Which of the following bacteria is the primary cause of necrotizing fasciitis? Streptococcus pyogenes on there, and also can cause strep throat. One of the virulence factors produced by group A streptococcus is blank, which can kill neutrophils and the immune systems of the first responders. It has this really nifty chemical that it produces called streptolysin S, and that you would have seen from the video and also from your book. Part D, blank is an anti-phagocytic factor and helps Streptococcus pyogenes attach to the nose and throat cells, and that is C. The M protein is what allows for that to take place and that to happen. Part E, what percent of patients are estimated to die from this disease? About 20%, and that's just from the video as well. Why is necrotizing fasciitis so difficult to prevent? Well, it's actually part of your normal microbiota. So if we did some sort of broad spectrum antibiotic against it, it would be difficult to erase all cases of it. Part B doesn't make sense. Mosquitoes are almost impossible to eradicate. Mosquitoes have nothing to do with the transmission of this. It is not susceptible to most antibiotics. It totally is susceptible to antibiotics. If this little girl possibly may have been treated earlier, there may have been a change, there may not have been. And D, it forms endospores. Streptococcus pyogenes does not form endospores, so only correct answer is letter A. And then on to part A, in which of the following cases would you suspect botulism? So we're moving on from flesh-eating bacteria to botulism, and there are different types. We can have wound botulism, we can have cutaneous botulism as well, and we can have food botulism, and we can have infant botulism when your little honey eats honey. That's why they say no honey for your honey until they're about a year old. They just don't have the immune system to deal with if they encounter this this bacteria under a year old. So with wound botulism, what you would expect to find, and these signs and symptoms would be just right out of your lecture and right out of your notes in part A. A person injures herself while gardening. 4 days later, she begins to feel weak and dizzy. And remember, you would expect to find this botulism a lot of times when people encounter it in the wound type of it. That's because they're gardening or they're digging in the dirt and they get really deep into the soil, and they may nick themselves as they do that. So being in a car accident is not going to cause it. Cutting vegetables at the dinner table is not going to cause it. Stepping on a rusty garden tool is not going to cause it either. Part A, are Clostridium botulism vegetative cells required to be present for food botulism to occur? No, because food botulism is not actually caused by the bacteria, it's caused by the toxin. So botulism is caused by the botulism toxin produced by the bacteria, not the bacteria themselves. So that's a very important aspect about foodborne botulism that I want you to understand, that it's not the bacteria that's causing the issue, it is the toxin that's produced. So toxin, toxin, toxins. Part A on page 5, all the following methods of food storage and preparation, all of the following methods of food storage and preparation, except which of the following can prevent foodborne botulism? Storing canned foods at room temperature. So that's not a method that's going to prevent botulism. Storing them at acidic environments, heating canned foods to at least 80 degrees for 20 minutes before consuming, storing canned foods in the refrigerator, all of those will. Part C is the one thing that will not prevent botulism. Moving on down to page 5, a cough can kill. A jumbo jet finally takes off from the airport in Johannesburg, South Africa, and Lance relaxes in his business class seat. The next stop, 16 hours later, will be in Atlanta, Georgia. Lance has just spent 6 weeks interning with a South African law firm that specializes in human rights issues. His work took him to clients living in crowded conditions in several poor townships. Now Lance's thoughts turn to the upcoming fall term, when he will practically live in a study corral. Law books his constant companions. The deep coughing of a woman sitting beside him interrupts his review. He glances at her. She is thin and huddles against the armrest, looking pale and tired. She often rubs her chest as if it hurts. When the flight attendant offers the woman something to eat and drink, she turns everything down. Her loud-infested coughing continues for the entire flight, interrupting Lance's sleep. 4 weeks later, Lance opens a letter from the State Department of Health and learns that he may have breathed in a potentially deadly bacteria on the flight from South Africa. The letter advises Lance to have a physician administer a skin test to determine if he has been infected, but it also tells him that he must wait 8 more weeks before being tested. Part A, an appropriately stained smear was prepared of sputum obtained from the sick woman on Lance's flight. Using oil immersion magnification on your microscope, what would you observe that would aid in the diagnosis of tuberculosis? So this lady had TB, consumption, as the oldies call it. And so what we're looking for, remember that tuberculosis doesn't gram stain. What we do is an acid-fast stain. And what type of shape are the Mycobacterium tuberculosis? They are rods. So acid-fast rods will be what we're looking for. Part B, the BCG vaccine is available to immunize people against tuberculosis. Had Lance been vaccinated, he would not have been concerned about being affected on the airplane. Why was he not vaccinated on here? And this is something that you would like to kind of cancel these things out, read these things in your book. But A, the vaccine requires repeated booster shots because the immune protection is short-lived. That's not true. It's not short-lived. The vaccine provides immune protection only for a low percentage of people. That also is not true. Pretty much everyone that gets vaccinated against it will have some immunity. Part C, the use of the vaccine in large populations results in the emergence of drug-resistant strains of Mycobacterium tuberculosis, and that also is not true. D, immunized patients have a positive skin reaction when tested, even if they have not been infected with Mycobacterium tuberculosis. Such false positives hinder the work of epidemiologists trying to track the spread of the disease. That one in there is a correct answer on there. So that's why we don't have these widespread vaccinations on there, because to determine what a positive result is for someone who actively has a case of tuberculosis would become much more problematic for epidemiologists. Part C, Lance's physician administered a tuberculin test to determine whether or not Lance had been infected during his flight. How was the test performed? And if you all, well, actually not if, you guys all had to have a TB test in order to register for class. So what they look for is they put a small amount of the cell wall material underneath the forearm of the skin, and they look to see for if there is a reaction, which means that you have antibodies against it, which means you fought the infection or you're actively fighting the infection. And how is tuberculosis transmitted? Well, what was he so upset with? Was he worried about sharing dirty needles with the lady on the plane? Was there a fecal oral route talked about? No. Aerosol droplets? Yes, because she was coughing, and definitely we're not talking about passing it in pasteurized milk. All right, so the next one in here is about Scott, who's excited to start college. He's looking forward to living on his own for the first time, new friends, taking courses. However, he's a little apprehensive because of his new community, and people don't really know him. He's a little self-conscious about the acne on his face and his back. He's always had a couple, but over the last few months, it's gotten a little bit worse. He washes his face a lot, so he's pretty clean, but still the acne seems to be getting worse. He tells his mother that he's scared to start college with his skin this way. His mother assures him that it's not as bad as he thinks, but she makes an appointment to see the dermatologist, and he learns that he isn't alone, that about 80% of teenagers suffer from acne. He explains that his acne is caused by propionic bacteria acnes, and its interaction between bacterium and Scott's immune cells that are leading to the acne. Part A, the name propionic bacteria is a term derived from the fact that the organism produces propionic acid during fermented metabolism. Which of the following statements about fermented metabolism is true? B, fermentation produces less ATP than aerobic respiration. However, it generates those electron carriers, NAD, which can be used in glycolysis. And this is just something that you probably remember from our discussions on it in the previous chapter, not in chapter 10, but in chapter 5, I think it was, that we talked about microbial metabolism. Part B, part of the reason Scott's acne is overproduction of sebum. Scott's initial instinct is that shutting down the production of sebum will be the quickest route to eliminating his acne. However, sebum actually plays an important role in maintaining the skin as the first line of defense against pathogens. Which of the following best describes the role of sebum as a first line of defense? And the last two bullets on that one. Sebum helps to keep the pH of the skin low, which inhibits the growth of microorganisms. Sebum forms a protective layer over the skin, so those two work. And then part C, acne is the result of inflammatory response. In this case, inflammation is resulting in acne that is very painful, and that's physically and emotionally for Scott. However, inflammation is a critical component of the normal immune system. What are the functions of inflammation? The second bullet, destroying and eliminating an invading pathogen. Third bullet, walling off the site of injury. That's the whole swelling aspect of it. And then the final bullet, repairing tissue damage resulting from inflammation. So here are your correct answers for page 8 there. And then for part E, which of the following outcomes are from activating complement? Optimization, which means it makes the bacteria or the target cells more sticky. Inflammation, kind of sounding the alarm. Inflammation is not comfortable, but it does have some really useful role. And then C, cytolysis, or the very last one, cytolysis. Part F, Dr. Smith assures Scott that although inflammation may sound scary, there are several possible options for treating his acne. Which of the following would be the most appropriate? C, an oral antibiotic such as erythromycin. D, clear light therapy. And then F, topical treatment with a product containing benzoyl peroxide. And in fact, most of your over-the-counter antibiotic or acne treatments contain that. So for page 9, all the way to page 15, and here's page 14 stuff, actually. So from 9 to 14, I want you guys to answer those questions on your own. And you'll be able to do that, to answer those questions on your own. And the reason for that is because we've gone over this information, and I want you guys to kind of practice a little bit. All right, so page 9 on to page 12, I guess page 12. All right, so going to page 13, you can't really see this gram stain really well, but it's the letter D for gram-positive cocci. That's in page 13. Select all answers that apply. It says based on the morphological arrangement to which genres of these bacteria refer to, whether cocci, and they're small, so micrococcus, and staphylococcus, they're in clusters, not in chains. And then here is the correct answer for on page 14. We have that up for us. And then on page 15, about Mandy, what she has here, it's C, coagulase positive, and facultative anaerobe, and that's what you'd find that just from reading the answer, reading the thing. So about Mandy reports infectious disease as being gram-positive, so the physician can act on the course of treatment. She inoculates the agar plate to grow the bacteria for a coagulase test and any other tests that might be needed. And we talked about being coagulase positive and being coagulase negative in our lecture class about how we can use that as ways to differentiate Staph aureus from Staph epidermidis. Other tests may include a fluid thioglycolate medium, which looks at oxygen requirements, and lastly, a plate of antibiotic disc is used for sensitivity. She then goes home for the day. The next day, Mandy is back in the lab. She takes a control slide with 2 drops of saline and a test slide with 2 drops of saline and a loop full of bacteria in the agar plate. She mixes the rabbit plasma onto each slide, then rocks them gently for 10 seconds. She examines the fluid thioglycolate medium and the coagulate test results. The test results show, and the test results are on your previous page there, where we're looking at the bubbling on the slide and whether or not there's been congealing happen in that test tube, and there was congealing happen. So C, it's coagulase positive, and looking at where those microorganisms are found throughout the tube, it's a facultative anaerobe. Compare the results of the classification scheme and determine the infectious agent, Staph aureus, because it is coagulase positive. And then here is that test that you're looking for: A, the bacteria, B, the zone of inhibition, and C, the antibiotic disc. And then on the very last page, on page 16, we have identify the TE disc on there. And when we look at the TE disc, is it effective for this particular type of antibiotic? So where we'd find the TE disc is letter A, so right here. And no, it's not effective, so that would be false. Alrighty, so there are chapters 19 and chapters 10.